Well, thank you very much for the introduction. Thank you for being here for what is going to be a very exciting panel today. We have illustrious panelists that will look at cancer progress from very different perspectives. But let me brief introduction. I'm Esteban Pombo, is Chief Technology Officer of TargImmune Therapeutics, where I have been for the last 7 years in a variety of roles. And I'm very pleased to introduce my co-chair, Marc.
Thanks, Esteban. Welcome, everyone, and thanks to the panelists for joining us with the panel today. Very briefly on myself, so I'm Marc-Olivier Turgeon. I'm a senior associate at Forbion. We're a well-established life sciences fund here in Europe, but as of last year, we can also call ourselves a transatlantic fund with our opening of our office in Boston and 2 new partners joining the firm. I just wanted to share a quick stat that could be interesting to this audience to show the interest of oncology for a lot of life sciences investors, but particularly for Forbion. And that's, we did an analysis recently on our deal flow, and out of 1,500 deals that we review every year, about 2/3 of those are oncology focused. So it shows that oncology is important. We see a lot of exciting deals, and also we learn a lot along the way. So
Great, thanks a lot, Marc. Andreas.
Yes, my name is Andreas Pahl. I'm the CEO of Heidelberg Pharma, and I joined a couple of years ago as a Chief Scientific Officer and built an ADC platform. And the core expertise of our company is a new payload. Probably we'll focus later on the ADCs. But this is a very different payload, a new mode of action, never been used in any chemotherapy. And then we also started developing assets based on that new mode of action. And last year was exciting because we are now advanced clinical phase 1. We already have seen objective responses, complete remission, so really clinical validation of a new payload. And I believe the world is looking for that.
Great, thanks very much, Andreas. Peter? Bryan.
Bryan Kobel, CEO of TC BioPharm. I think it's the first time I've been referred to as illustrious, by the way, so I appreciate that. We're an allogeneic gamma delta T-cell company, so cell therapy. Of those words, 5 of them are apparently are curse words today in oncology and investing, but we have a phase 2b ongoing in acute myeloid leukemia. We announced last week that our cohort A in relapse refractory has been completed. We're scrubbing data as we speak, and cohort B, which is minimal residual disease patients, is ongoing as we speak. It's donor-derived, so universal, and we think we can treat not just a number of oncology indications and patients on a broader basis, but also we're expanding into infectious disease this year as well.
Very exciting. Thanks very much, Peter.
Yes, thank you. Peter Llewellyn-Davies. As you can hear from my name and accent, I work for an Austrian company called invIOs. We develop cancer drugs, and we are currently developing a cell therapy, autologous cell therapy, ex vivo, with a turnaround vein-to-vein time of 6 hours, unheard of, where we've completed phase 1 trials, and also a small molecule with this dual-track approach. We're working with the Dana-Farber Cancer Center in Boston to treat glioblastoma and other indications. My personal background, I'm not a biochemist, so I think I'm the only one here who had the worst degree or the worst marks in biology at school. But joking aside, I've been in biotech for about 20 years, and I've been fortunate enough to work for over 10 companies, either as a CEO, CFO, or CBO, and done quite a few deals from monoclonal antibodies to vaccines to TCRs, cell therapies, or imaging agents. And my background is probably the experience in oncology in respect of manufacturing and commercialization.
Thank you. Robert?
Hi, everyone. I'm Robert de Jonge. I'm the SVP and Head of Corporate Development at Mestag. Mestag is an immunology company, so we're focused on the interaction between the activated fibroblast and the immune system. Our lead program is an oncology program where it's a lymphotoxin beta receptor agonist, which is targeted to the tumor. And we're able to induce tertiary lymphoid structures in solid tumors, which is a really exciting new piece of biology. Tertiary lymphoid structures are basically germinal centers that act like lymph nodes, but are operating in the tumor itself. They provide access to lymphocytes, to T cells, as well as they prime or educate T cells locally in the tumor, and they boost responses. And we know that in those patients that are responding to checkpoint, but also to chemotherapies, that those patients often have TLSs. So very exciting program. The second program that we have is an immunology program. And again, this is an antibody-based therapy where we're dampening down myeloid cell-driven inflammation across a range of inflammatory disease indications, including inflammatory bowel disease, Sjogren's, and lupus. And when I talk about myeloid cells, this is monocytes, macrophages, and neutrophils. My background is originally molecular biology, and I've started my career in venture capital. For the last 12 years, I worked in business and corporate development in venture-backed biotech companies.
Yeah, thanks everyone. Before we dive into the subject and the discussion, we wanted to provide a bit of context because obviously cancer progress can mean so many different things. And I think one of the thing that's great about cancer innovation is that there is a lot of innovation that works. And over the last decades, we can see a lot of progress and that translates into survival benefits for patients. And so we've tried to anchor the discussion here a little bit on things that have worked and looking at the key pillars of cancer treatment. Obviously chemotherapy, radiotherapy, but now recently over the last few years seeing how this is translating into ADCs and BioPharm that have a more precise way of working but using these kind of similar mechanisms. Obviously IO, and we have a lot of expertise here at tackling IO in different perspectives. And obviously looking at precision oncology as well. So to kick it off, maybe as a first question, given all the progress in the ADC space in the last few years, maybe we start with that. And first looking at obviously the progress of HER2, maybe going into earlier lines of therapy and maybe replacing chemo in some context. So maybe Andreas, you can tell us a bit your perspective on whether our ADCs really replacing chemotherapy? Are they here to stay, or is this still just another wave of hype for this type of modality?
Of course, I'm a little bit biased working in an ADC company, but I believe the ADCs are the modality which are not only active in heme, but also in solid tumors. This is a little bit in contrast to the CAR T cells, the T cell engagers. So we truly believe that they are coming and staying and will even expanding in solid tumors. And the HER2 they each year with a tremendous success is very clear that they will make their way in the solid tumors in order to stay there. The other point was your question.
Maybe we can continue on now that this is getting assuming that it's a modality that's here to stay, then what are the way to differentiate in this indication, and also invite the other panelists to comment as well on our ADCs here to stay and from a different perspective as well.
No, I forgot the point was for first line. It's really becoming clear, let's say, from the patient perspective, because you have a much broader therapeutic window with ADCs compared to chemotherapeutics. And you see trends with some of the ADCs already moving to earlier lines. You see this in the heme-onc space. Blenrep is trying to do this, the GSK, because from the patient perspective, you want to have a therapeutic window, which you don't have with a naked chemotherapeutic agent. The downside probably, let's say, from a payer perspective, it will be a challenge if ADC is moving to earlier lines, but from the patient perspective, I believe the trajectory is very clear. Let's say the learnings, it has been challenging, and it's quite, the universe of payloads is pretty limited. We have seen the era of the tubulin inhibitors, then we have seen the era of the DNA damaging, the PBDs, and each have specific side effects. And it takes quite some time until you learn what are the specific side effects, how you can deal with your class of payloads. And that's why the diversity is still very low for the ADCs in general with regard to modes of action and diversity.
But there's a lot of excitement about just putting something radioactive and throwing it there and pretending it's an ADC. What is the panel's experience, and what do you think about this relationship between radioactive antibodies or radioactive ligands that have really nice specific effects, radiotherapy, localized targeted radiotherapy, if you want, and the ADCs? Are they complementary? Can you use the same target with both payloads? How do you see things going forward there?
Maybe I can start briefly, but I don't want to occupy all the time. So our view of what we experience, they are more complementary. There's still, let's say, issues with the supply chain Also, the number of hospitals, it's not for the outpatient setting, so it's a little bit limited. What you see from the radiotherapy, also the side effects. So we truly believe they are complementary.
So we see that there's really a technical issue, and manufacturing is critical. I know Peter, for example, has an opinion about feasibility. What do you see from that perspective between ADCs and radio ligands or other approaches? Because of course, cell therapies and are also not entirely trivial to manufacture. Would you like to comment on that?
Yeah, sure. I mean, it always comes back to the same thing. What's the commercial model, right? And we're all scientists here and everybody's excited about what's happening. But in the end, these non-successful ADCs, let's say the failure of the AstraZeneca Daiichi project, and et cetera, these are toxicity issues, right? And toxicities are a big issue with ADCs, and that needs to be solved. And how will it work in the real world? So from personal experience, what's the route for manufacturing? How do you get this to the patient? How is this going to work out commercially? If you find out you're going to be in an indication where you're not going to make any money, then you won't get the money in the end, and you're spending a lot of money just following a trend that might not in the end follow through in a commercialized or regulatory approved product.
But it's probably not just a trivial story that you have a target, you make an antibody, the antibody fails for safety or efficacy reasons, then you put something to punch it up and then increase the efficacy, hoping that the safety doesn't suffer. Then you are successful. In many cases, you're not. The ADCs suffer from unexpected toxicities or expected toxicities of the payloads. And then you say, okay, well, let's try a radiochemical, but we may have to tweak this a little bit. Or then let's try bispecific. Let's try and recruit the immune system by bringing the old antibody, or at least part of it, together with something that binds a cell, and then bring the whole thing together. So maybe in that perspective, am I just making a caricature of this evolution of target space, or do you think that there's something we can learn from this analogy?
But just to finalize my comment, I mean, there's a saying that says that the antibodies, the monoclonal antibodies that don't work, turn into ADCs one day. I'll leave it at that.
Thank you.
Yeah,
Any other thoughts from Robert?
I thought maybe what's interesting as well is that Andreas just talked about the therapeutic window, which I think is very important in this particular context, right? We've seen the ILD, so interstitial lung disease, come up with some of those ADCs as well, and that is, of course, a problem. So I would be quite interested. So beyond the safety point, I think there's another point to make, which is around response rates, because the initial response to ADCs is actually pretty good. It's reasonably high, but then the duration of response with some of these ADCs is actually not that impressive. So I think there are two things that need to be solved. Would love to hear Andreas's view on that.
So let's say we are in multiple myeloma, and the observation is that the response rate is not so much different compared to bispecifics, ADCs, and CAR T cells. The response rate is higher for these modalities.
Sorry, I should specify I was talking about solid tumors.
Okay, yeah. Solid tumors, that's of course is an issue, but I believe in general, because solid tumors in general have a very poor duration of response. Again, CAR T cells, the T cell engagers does not really work up to now, and that. But solid tumors sticks, and is still, we're talking about cancer progress, the biggest thing to go for in this space, because there is really a lack of duration of response and response rate.
I think maybe we switch to a next topic, and again we kind of segue into this a bit with the durability of response. I think one field where one area of oncology where we've seen durability of response is certainly IO, and that is true with checkpoint inhibitors, but also with cell therapy. So maybe on the IO. Well, we've also seen like checkpoint inhibitors being well established. The summit data has certainly been on the lips of a lot of people with the bispecifics with the VEGF. But I think when we think about cell therapy and IO, we also think T-cell engager cell therapy. So maybe I'll invite some of the IO experts here to comment on how do we see cell therapy versus T-cell engagers in the IO going forward.
Well, yeah, I think that I was very impressed by the T cell engager data. So we know that we have the T cell, the DLL3 T cell engager by Amgen that was recently approved, and obviously people are getting more excited. And whenever we speak with pharmas, but also investors, there's a lot of interest in the T cell engager space. So I think those are coming through. I think the other point maybe to make, so on the CAR T cell therapies, in solid tumors specifically, I think people are now moving more into that, but I don't think we have seen a lot of clinical data yet to show that that is working. I think one aspect maybe to point out is that I saw the AstraZeneca data on their GPC3 CAR T cells that at ASCO last year, and that showed some responses in hepatocellular carcinoma, so that's great, and I think others are moving into that space. So, but the problem to crack there is, can we actually have the T cells, A, getting into the tumor where they need to be, and B, can they actually perform their job in this immunosuppressive environment?
Yes, Bryan.
Yeah, I mean, I think that's an interesting point, right? Which is you're trying to engage a population of cells that in these really sick patients just kind of sort of doesn't exist, right? And,
sorry, Bryan, can you speak closer to the microphone? Thanks.
Sorry, apologies. We're saying that that's one of the issues they face, right? Is that it's an interesting comment in the sense that you're trying to engage a population of cells that is very much so hampered or just doesn't exist in some of these patients, especially when you start doing earlier stage trials. So the question about moving to earlier patients and treating them sooner when they might actually have a more constituted immune system and more constituted T cell makes a lot of sense on the T cell engager side. I think you're also seeing, an interesting sort of back and forth because you've got a lot of positive data in there, but you also have companies, Lava Therapeutics is a great example, that announced strategic alternatives yesterday. They've got, 75 million in cash in the balance sheet and they can't get out of their own way. So I think there's, for whatever reason, I think it probably gets back to Peter's comment around the commercial side of this, right? How big is your patient population really? Can you treat solid tumors? If you can't, everyone's sort of moving on. So, I think cell therapy and immune oncology is an interesting sort of space now, which is, what is the brass ring? Is the brass ring where a monotherapy for a solid tumor, but it's a small patient population? Or is the brass ring where a part of this solution, where a combination, it's T-cell engager plus something else? I think that gets back to how do you really take these therapeutics from where ADCs were 10 years ago to where they are today? So I think, and I don't know what the answer is, but that sort of commercial angle seems to be the dynamic.
Peter, do you have a comment on this?
Sure. So we're in immune oncology, and we've developed a cell therapy platform where we focus on treatment of solid tumors. We've had some good results. We've had some good preclinical results. We have some good clinical results. But the question is, of course, what sort of population you go into, what sort of indication you go into. We're going to a very difficult one, glioblastoma, because we think this will work, because we're using TILs here, tumor-infiltrating lymphocytes, and silencing CBL-B. So what we're using is a checkpoint inhibitor as a possible way of using cell therapy to kill off the cancer.
Thank you. So it's very exciting to see that, of course, if you can modulate the immune system to effectively attack the cancer acutely and generate a memory of these neoantigens to have really long-term remission, that is the hope of immunotherapy, right?
Yeah, no, I think this is an interesting point. So I think one thing that was just mentioned around the PD-L1 or PD-1 VEGFs that obviously everyone is talking about and following closely. I think that data, obviously, we're still waiting for the phase 3 in the US, but some of the phase 2 data also with BioNTech shows promise. So I think that the field believes that this is going to come through. But one of the interesting things I thought, which was an announcement actually earlier this week, combination of these types of therapies with ADCs. So I know that BioNTech is angling for that, but also Pfizer has just announced that they were working together with, or going to work with Summit on combining their PD-1 VEGF with an ADC. And I think that could combine the durability with the punch of the ADC in some ways.
I have maybe a follow-up question on this. I think it's an interesting point. I think that we see, and I think few people have touched on the combination part of it. if you're a small biotech and you're now developing a new agent in the IO space or a new ADC, how do you compete with big pharmas that are going to put all of their agents that are in their portfolio together? Someone like, if you look at AZ and the way they're developing their ADCs together with their own PD-1 or PD-1 bispecifics.
So Big Pharma has lots of wasted antibodies that they want to repurpose.
Is that a call for Andreas?
Of course, it's challenging. So usually you start with a monotherapy, your dose escalation, and then if you look, let's say, into the commercial pathway, like what Peter was saying, you find a niche probably for a monotherapy, but sooner or later you need to come for combo. That's no question. And this usually is without reach, let's say, for a biotech company, because these studies get so huge, so long, so expensive. So I believe one of the possibilities to dose escalation, show that your drug is working in a monotherapy, and then look for a partner who can, let's say, take over or make a joint development.
I think
Peter?
it
Sorry.
depends on if you're really
Right.
looking for like competition, right? I don't think small biotechs that are developing drugs have these grandiose ideas that they're going to all of a sudden become Genentech or Regeneron. They're going to fight against big pharma, right? So I think to answer your question, big pharma will always buy innovation, right? They bought Poseida Q4. They bought another company with an X, I forget the name of it, and within the last 90 days. They're not going to innovate themselves, right? So if you can create some type of a valuable product that they believe has some part of that portfolio that it can outperform, they'll always step in and acquire it, right? So that's, I think from our perspective, at least as a small biotech, why you're fighting the good fight every day, which is if we can innovate enough, whether it's an inch or a mile better, there is a light at the end of the tunnel, right? I think that's sort of the mentality.
Yeah, and we need to get in earlier. That's our personal view. Because quite simply, at the moment, when we're treating patients that are at a very late stage of their disease, and we're ending up having patients with a relatively low immune system or non-support immune system, and if it works in them, then it should work much better with earlier patients. Challenge number one. Why are we coming in late? Because others are there before us who we can't really afford what they're paying. So we're stuck as a biotech company. And that's the long-term story of us in general. So we have to find other innovative ways to get the product ready for market earlier by sorting out the manufacturing, for example, right at the beginning. So what we did, we went a different approach and said, right, let's do a feasibility study with our own manufacturing system and do GMP readiness, and went to the authorities, the EMA and the Austrian authorities, got their approval. So we said, right, we're ready to go for a clinical trial. If a pharma company then comes along and says, right, we're ready to go, what have you done on the manufacturing side? And that's certainly a bit of advice I'd give anyone. Make sure you have the manufacturing set up as quickly as possible and not just hope it will work out in the end.
So one of the aspects of great innovation in the field is the ability to come up with a drug that will sort of modify the response of the immune system to early disease and then prevent recurrence. So help in the neoadjuvant setting and then surgery and then prevent recurrence. However, that means that you probably have to do a head-to-head trial against classical chemotherapy, for example, for breast cancer. And that is extremely difficult and expensive unless the results are super magical. And even then, regulatory hurdles will be there. How would you approach that?
I guess hope that the FDA misses it because there's so few people there that are left. Have they fired everybody that you can just sneak one past them?
They have forgotten about the old drugs because none of the new old people are there.
I think it is a good point, right? So I'm just thinking about clinical development. So we're about 14 months away from the clinic with our oncology therapeutic. And when you look at like typical trial designs, obviously you need to start in end stage patients. And I think that brings with it certain challenges because these are patients that maybe had 6 lines of therapy. They have a completely burned out immune system. And obviously everyone knows that the phase 1 dose escalation is set up to look for safety. But as a small biotech, the thing that everyone wants to see, especially if you speak to investors, is some signals. If you don't have it, then I think you have a big problem on your hands. So it needs to be able to do both. So one of the things that I think is quite challenging is that that it kind of there is the tendency to start in those end stage patients, and if there would be more possibilities to start in earlier lines of therapy, I think that would be an interesting prospect. I know there's initiatives that try to do that. There's one in the US as well. There's also challenges with that because you have a different treatment or you have longer treatment duration possibly needed to see efficacy. But I think it's an important point.
I think to your point also on the early stage and how do you compete against chemo and things like that in an earlier stage patient relative to a trial, that is potentially a viewing through an old paradigm, right? You have machine learning, you have AI coming on. There might potentially be an application to clinical trials where you don't have to run these side-by-side dynamics and things like that, right? Because you should be able to, with that technology advancement, extrapolate out a bunch of different things, right? There's a ton of data on chemotherapy, right? So it shouldn't, not that it can today, but at some point that wave is coming where AI should impact these trials to undercomplicate them.
So you could have like a digital twin or that do a head-to-head comparison in silico, and that would be acceptable. But of course, the hurdle for that is still very high.
Oh, of course. Yeah.
And we're really looking forward to something like that because right now, as you quite rightly said, Robert, quite rightly said, we've got very late stage patients, right? So we treat them, they respond very well, but then they pass away. Not necessarily because of us, not so necessarily because our system didn't work, but they're too far away. But if we were able to follow up with them for 1 or 2 years and see what the long-term response is, those patients who had the long-term responses and survived for 2 or 3 years, they're the ones who show real efficacy, right? So this is the challenge, getting earlier on in the indication, early on the treatment regimen, and then making sure we have biomarkers, follow-up data, and have that ready.
Maybe a question that kind of ties in a bit the IO and the ADC landscape, which is we were discussing before the panel on new targets in oncology, and that's always a big topic. So are there enough targets, and are we competing for all those targets, or is there a key to look for new biology? And I think also Robert on the IO side of things is also looking at new biology from a mechanism perspective. I think that's something that's important for Mestag, right?
Yeah, it is, and I truly believe that it's important to work on novel biology and novel targets and bring those forward. Obviously, we with our lymphotoxin beta receptor agonists are doing exactly that. So we're working on a new piece of biology that only came out fairly recently, and we're able to leverage that to boost the immune responses. But I think also if you think about the ADC side, what you do see a lot, and I'm sure my colleagues here will be able to comment, is there's a lot of aggregation around the same type of target. So I think even there would be a benefit of further innovating on that front too. But I definitely believe that going forward we need more on both sides really. And then ultimately I do think that combination type therapies would make a lot of sense mechanistically in this context.
People have been doing one drug for a target, and then a me too, and then a me better, and then unimproved. I mean, that's been classical pharma development strategy and competition. And at the end of the day, you have a number of drugs, and really they behave differently in different patients. So I think there are an advantage, but of course it's very difficult because you have to demonstrate benefit, right? And how would you do that, Andreas?
Yeah, I have a little bit of a different opinion on this one because there's, let's say, the big pharma, or let's say the world is focusing on quite a few targets, but they are clinically validated. And finally, if you look at the resistance of the patients, they mainly become resistant to the mode of action. There's some target loss, target downregulation, but majority is still resistance to the mode of action. So we have new biology, a new mode of action, and there are a lot of examples that you can sequence on the same target with different modes of action. It can be different payloads on an ADC, but you can also sequence a CAR T cell and an ADC unless the target is stable. And there's a lot of data in the ADC space that even, let's say, if you have TROP2 as a target and you have different mode of actions, you can sequence this. So I firmly believe that biology, new modes of actions, whatever that is, let's say, key for overcoming resistance and to have benefit for the patients.
And that's the interesting thing about checkpoint inhibitors, right?
Yes.
Because they don't define the mode of action. Silencing them opens up various kinds of modes of action.
Yeah, and of course, you need an activated immune system for the checkpoint inhibitors to work. So you also need for the drug to get to the tumor and the immune system to be in the vicinity of the tumor and activated. But maybe Mestag has a couple of ideas about this macrophage stuff.
Yeah, I mean, we just talked about it already, but we believe that our drug is able to achieve two things, right? One is facilitating access to the tumor, so we're able to induce so-called high endothelial venules, which are specialized structures in the vasculature that allows lymphocytes, T cells, to get into the tumor, which is one of the, I think, important components here. And you can think about that in multiple ways. If you want to combine it with things like T cell engagers, they also require the cells to actually be there. And the other component that we're able to do is to induce these beautiful germinal centers, which are called tertiary lymphoid structures. And in those centers, you get the same thing as happens in the lymph nodes, basically priming or education of new anti-tumor T cells. So there's no travel involved, which I think is nice. But that allows, if you combine that with agents that generate more antigen, so you can think about ADCs, but also chemo, you can think about radio, you can actually use both of those mechanisms to get more cells into the tumor, but also to get more anti-tumor T cells educated locally, which I think is very exciting. So that's how we look at the world.
And that takes you to the next step, right? Rather than try and do this sort of endogenous education of the cells, why not just generate a vaccine with an mRNA vaccine? Like there's an interesting total tumor RNA vaccine that has been shown by the University of Florida to have some very exciting data in animals in early stage in humans. That kind of an approach is very complementary to the drug-induced neoepitope and so immunity, and you don't need the travel. So there's the travel and the immunity, right? Those are two related aspects.
Yeah, I certainly think that the vaccines are a way forward, but as a monotherapy, we're a long way away. But perhaps as a combination therapy, with immune therapy, for example, that could definitely enhance tumor killing.
Do you have a comment, Andreas?
Yeah, maybe, John, because we are talking about, let's say, individual therapies, but let's say in the history of cancer, it was always a combination. The treatment of solid tumors is usually a combination and not a monotherapy. So I believe the challenge is you have so many things on the table, some different modalities. Finally, what is the best combination and will be probably different for each patient. So I believe the challenge, you're talking about AI, so we talk about biomarkers, so you need to find the right combination for each patient. I believe that's the biggest challenge.
We've been talking a lot about IO. I think one of the obvious thing in the last couple of years is probably that the interest from investors in pharma towards IO and new approaches seems to have maybe reduced to a certain extent, and maybe I'm part of the problem. But essentially, what is the panelists' view on that, and how do you continue to innovate in this kind of environment where the late stage clinical failures have led to this kind of climate? But it would be great to hear the thoughts from panelists.
No, I think you're absolutely right. So I think that if we speak obviously on a regular basis with investors and also pharmas, and it's very, as we say in the UK, very Marmite, right? So in some cases, when you speak to investors and you mention the word oncology, they almost immediately close the door. But in other cases, they see that actually a lot of value in their companies historically has been driven in M&A, also in the oncology space. So they tend to still be able to tilt their portfolio towards the IO or towards the oncology space. I think also that a lot of people have been burnt, and we've done a lot in the IO space with failures in TIGIT and many other related checkpoints or underperformance of some of those checkpoints. So it's not unusual that people will get a bit discouraged by that, but I don't think we should be really. And I think what also has been happening is because the T-cell engagers and the ADCs were fairly new over the last couple of years, that a lot of the investors have focused on placing their bets in those areas. And now actually what we see now is that they are more open to listening to other types of stories, which I think is important. And as I mentioned before, if you think about the PD-L1 VEGF, which I would firmly classify in the IO space, and I just had a quick look at the Merck Keytruda revenue of last year, and that sits at the $30 billion mark. So it's quite high. So that tends to draw the investors' attention into this area again. So if we get approvals and positive data there, my belief is that will shift, and we can already see some of that happening.
Mm-hmm. Plus, in spite of that revenue, there are many patients that don't respond. That would be,
exactly.
Technically would be, I mean, we should respond, but don't.
Yeah. I mean, I think the answer is when you're going through hell, keep going, right? And that's kind of where all these companies are, just the sort of reality. But I don't think it's just the failures clinically, right? I think one of the bigger issues, and Peter touched on it, is the commercial model, right? How do you create a real economy of scale, especially in the manufacturing side, and get to a stage where you can commercially manufacture a lot of these IOs and reach a broad audience instead of being extremely targeted, very sliver oriented. It's a small market, and you're going to charge people $2, $3, $4 million for the treatment, right? That's just, it's never going to work, right? So, and you're seeing that with Bluebird Bio or whoever they are now, whatever they get renamed as post being acquired, or whatever that was. So I think that's the next big step for the IO space, is how do you really create an economically viable business or model around these technologies? I don't think it's just people don't think they work. I think people see the value of, using the immune system, right? It's there for a reason. It's developed for a reason. It's evolved for a reason. You're seeing people shift into how do we create a healthier system, right? How do we do things that are less edited, less toxic, right? So that is a way forward, at least in my opinion. But I do think there's a lot of issues that need to be solved, and you see that happen in all biotech cycles.
Yeah. Robert? Yeah.
Yeah, sorry. I think the other point, maybe to point out what Andreas was saying, is I think it's important to also make sure you know which patients you need to target, right? So if you know that there's a specific patient population you want to go into, that you basically focus your plan to get to those patients as quickly as you can to establish proof of concept.
And we're moving in the right direction. We're getting to know the patients. We're getting to know the disease better and better. We're getting investors who are not just following the trend, but they're following the long-term scientific rationale behind the company's approach. And through that, we're getting new targets, new pathways, and we're getting brand new ideas. And we've got to follow the science and not the trend. We've got to stick at it as companies and just ignore, because every time in my career, I've followed what the investor said. 2 years later, he said, yeah, but my opinion's changed. So keep going, guys. And that's what we have to do. If you run out of money, just try and keep going anyway. So
great. Maybe we open to the audience. I see a hand being raised back there. No, somebody was just scratching their head. Now, if you'd like to share your hair scratching with the rest of the audience. Yes, please.
I guess it was Robert who talked about the Keytruda revenues. What happens when the patent expires? And what do we look for? What do we expect the PD-1 sector to look like once these patents start expiring?
I think there's one point maybe to make. So, of course, the pharmas have not been sitting still. So you're probably familiar with the fact that they have developed subcutaneous dosing, which has been approved last year. And what you've seen with the Tecentriq market approval and basically their launch in the UK, 30% of the patients has actually switched from IV to subcut. So I think if you look at the analyst reports and the predictions that, for example, BMS is doing, is that between 30 and 50% of the patients will move on to subcutaneous dosing. So I think, of course, they have protection over that. So that's an area that will definitely grow. I'm not saying that it's not going to be a problem for Merck when their patents expire.
But it's clear that the focus on improving the patient's experience during therapy is something that will help people to continue innovate in the field.
And I think to cycle back again to the summit data, right? The PD-1 VEGF, although this is still early and we don't know how it's going to turn out, I think it's kind of given a bit of hope, and it's a bit of a coincidence, but in some way a good timing for pharma company that these type of mechanisms are now coming as improvements on the PD-1 franchise.
I agree, and I think you may have seen as well that Merck has also acquired or licensed a PD-1 or PD-L1 VEGF antibody, bispecific antibody as well. So I think all of the pharmas have stocked up on the PD-L1 or PD-1 VEGF to be ready if the data is positive for the next phase. And there is real benefit if you look at the early clinical data between the two. So that would be, at least my opinion, if it comes out positive, that would be the next phase.
So no further pertinent questions from the audience. Let's go back and yes, one more, please. Yeah.
Please hold for microphone.
Thank you. I have a question about cancer vaccines. I think this is one of the areas which was overlooked, and I think now everybody is waiting for BioNTech and Moderna when they will provide their readouts in the next couple of years. But maybe you have any thoughts whether personalized cancer vaccines or off-the-shelf cancer vaccines has its future?
Let's say my personal view, it goes on for years and years and years, and the promise still needs to be fulfilled. So again, the question will be, we're talking about a monotherapy, and the dream that you will have the magic bullet who cures cancer, I don't believe this will come true. Personal opinion.
Mm-hmm. I think that the early vaccine data is looking interesting. I think there is confirmation required, and that's what everyone is waiting for. But if that is positive, then obviously this will become a new area as well. I can't judge how good the data is or not. I'm not an expert in that particular area.
Yeah, and in combination with immune therapy, I think it could be a future treatment.
We're getting close to the end. I think we haven't covered obviously everything, and then apologies if people are working in precision oncology and we didn't get a chance to get to these topics before another time. But I think to wrap up the discussion, I think we wanted to ask each of the panelists where they see the biggest unmet need and opportunity for innovation in oncology in the next few years. So maybe starting on my left to Andreas.
Yeah, but let's say in line what I've said, I believe new mode of actions, whether these are payloads for ADCs, new modalities, but new ways of killing tumor cells. Because the typical journey of a cancer patient sooner or later becomes resistant. He needs another line of therapy, which needs to be different to compare to all prior lines of therapy. So we are working in the fields of new payloads with new mode of actions, but there can be new type of IO agents, cell therapies, whatsoever. And what we observe in tumor, patients already survive much longer as compared to 10 or 20 years ago. So the journey is longer, but again, the demand and the medical need for new lines of therapy with new mode of action still continues.
That's a tough question. I think if I knew it, I'd be retired somewhere. What I think the next big step, or at least I hope the next big step is to your point, turning these diseases that are acute and have short lives and very low durability response into sort of a chronic disease. So what's the combination process you have to deliver, whether it's ADC, chemo, whatever, with something else? And maybe these patients aren't necessarily in remission forever, right? But they're given a treatment that's recurring over the course of time, every 6 months or whatever it is, to where they can live a full life, right? How do you keep that quality of life high, which I think is the other side of this treatment, which is their quality of lives go to basically zero for a period of time. So interestingly enough, I think it's a combination of how do you extend the durability of response and how do you do that in a way that keeps the quality of life high?
I think maybe a quick question on this before we continue. We discussed a bit earlier on the safety aspect of it, and obviously efficacy is always seen as the priority, but I think more and more we're starting to see therapies where they pay attention to the safety. So any thoughts on that as to like what needs to change within the regulatory agencies or treatment paradigm to kind of enable that increase in safety as well?
I don't know, right? It's quality of life, not quality of survival,
yeah.
Right? So if you keep somebody alive for a period of time, that's not exactly them living. I do think, to your point, there needs to be innovation on the regulatory side. I think a lot of and this is not their fault, right? They're overwhelmed. There's a tidal wave of information and applications and everything else. But there does need to be a change and a viewpoint and a shift and how they view, like for pancreatic cancer, right? If they keep you alive 10 months, all of a sudden it's a win. Even though the patient's basically hooked up to a bed for 9 straight months and has no quality of life and is barely alive, barely awake, right? That needs to change. There needs to be a heavier shift into taking into account the safety, the toxicity, and the quality of life these patients have. And that has to account for something above the efficacy to some degree. And that's why, to your point, I think hopefully we come back to that sort of level of immunotherapy, but really cell therapy, and how that is taking a healthy system and exacerbating on it and making it better and keeping your quality of life high.
Yeah, oncology is here to stay. The longer and the more time and money we spent on this, we'll get more information about the patients, personalized medicine. But we need to do 2 things. We need to lower the costs, whether it's replying to emails from Mr. Musk or not, but I don't think that will help. But we need to lower the cost, and we need to simplify and speed up the processes to get clinical proof of concept. I think getting clinical proof of concept was one of the biggest hurdles for a biotech company. And how can we solve this? We need to find better ways of doing this. And perhaps AI will help us in this respect, but we're a long way away from that. So simplifying the process and dramatically reducing costs to enable many more companies to further develop their projects.
Thanks, Robert.
Yeah, I already alluded to it earlier, so I agree with the panelists here. So for me, it's a new mechanism of actions, combination therapy is the right types. We just already discussed that, and also I would argue improvements in the regulatory landscape to get to earlier patients quicker.
Thanks very much. So I'd like to thank our panelists and really summarize some of what for me are our key takeaways from this discussion. First, we will see progress in cancer therapy. The progress in cancer therapy will be novel therapeutics, novel approaches, novel payloads. Some of the old targets will still be used. There will be improvements in the ways we use and understand the biology of the old targets to get new biology and get remission and long-term remission of cancer. There will be all these tools to try and simplify, accelerate, and improve the readout of clinical trials, and that we will be able to go into earlier stage cancer so that the benefit to the patient and the long-term quality of life is then much better than attacking only late-stage disease where there is limited possibility of actually helping the patients beyond a relatively short period of time. And of course, there's going to be neuroscience, and there's going to be a lot of excitement and investment in new technologies. So I do hope that with my co-chair, we'll see many new ideas and approaches. We will hear about regulatory agencies taking on some of these new technologies in AI, sort of digital twins, virtual clinical trials, and so on, that are just starting to be understood as what the possibilities are. It's something that's coming very strongly. Plus, the business model has to make sense. And you have to be able to present a proposition that is financially attractive for investors, so that these things are being funded. But if they're not being funded, as Peter say, just stick at it, because they will all come back. So thank you very much to the panelists. Please join me in giving a round of applause.